Hi, my name is David Parsons and I work for SLU in Umeå. This presentation is part of the ProRefine project, refined forage legumes as local sources of protein feed for monogastrics and high-quality fiber feed for ruminants in organic production. The specific topic of this presentation is using a leaf stripper in mixed stands of grass and clover. The project focuses on two methods to biorefine lucerne and red clover. Firstly, using a leaf stripper, and secondly, using a screw press. Both methods fractionate the forage, creating different products that can be used for different classes of livestock. This is an illustration of the leaf stripping method used in this experiment. As the leaf stripper is driven through the plot, it removes leaves and the upper portion of the stems of the legume, which are collected in the machine. The more fibrous legume stems and the majority of the grass remains and is harvested using typical harvesting methods. To give some background on this topic, red clover is the most commonly grown forage legume in Sweden. However, it's rarely grown as a monoculture and is typically grown with grass in a mixed ley, because mixed leys are easier to ensile and are less susceptible to winter damage. Mixed leys are typically dominated by grass in the first cut and contain more clover in subsequent cuts. So there's potential to use a leaf stripper opportunistically in mixed leys when there's a high fraction of clover. The project was therefore designed to answer the following questions. 1. Can a leaf stripper machine be used in mixed leys to separate red clover leaf from red clover stem and grass? 2. Does leaf stripping generate different concentrations of protein, fiber, and digestibility of the resultant fractions? And 3. What are the implications for feeding livestock? The specific machine that we used was the Prem Alpha Mini, which was designed to be used in experimental plots with the same technology as larger models. It has an electric drive and variable height, ground speed, and rotation speed. In June, July, and September 2021, we sampled mixed stands of clover and grass. We took various measurements, including plant heights, phenological stages, and measurements with sensors. We took samples so that we could assess the yield and quality of the different fractions. I'll explain these fractions. Before harvest, there were the clover, grass, and weed fractions. Combined, they made the whole sward fraction. From here, we'll ignore the weed fraction because it was a very small component. After harvesting with the leaf stripper, we then had the leaf stripper fraction and the residual fraction. Forage quality analysis focused on crude protein, neutral detergent fiber, NDF, ash, and digestibility. To address the first question, the leaf stripper worked well in mixed stands, removing on average 1/3 of the available forage biomass. In this picture, you can see the difference between the original sward on the top and the residual fraction after leaf stripping on the bottom. Here's an example of what the leaf stripper fraction looks like. It's predominantly clover leaves with some clover stem and grass. The results show the differences between the different fractions. The pre-harvest fractions, clover, grass, and sward, are green in this figure, and the post-harvest fractions, leaf stripper fraction and residual fraction, are coloured blue. The letters indicate whether fractions are significantly different to each other. If fractions share a letter, they're not different. For crude protein, as depicted here, the clover and grass fractions were not significantly different to each other. Following leaf stripping, the leaf stripper fraction had a higher crude protein concentration than all other fractions. The residual fraction on the far right had a lower crude protein concentration than the leaf stripper fraction and the clover fractions, but not lower than the grass fraction. For NDF, all fractions had different NDF concentrations. As we would expect, the clover had lower fiber than grass. The leaf stripper fraction had only a slightly lower NDF than the clover. The residual fraction had a lower NDF than the grass fraction, which suggests that the quality of the residual fraction is still high. These results suggest that the leaf stripper can effectively increase the crude protein and reduce the NDF concentrations of mixed stands. For the digestibility results, there were smaller changes and less differences between fractions. However, the leaf stripper fraction had a higher digestibility than all other fractions. So to address question 2, these results suggest that the Prem Alpha Mini was able to function well in mixed stands of red clover and grass, resulting in a product with higher quality than the pre-existing sward. Compared with the original sward, the leaf stripper fraction had 33.9% higher crude protein, 27.4% less NDF, and 3.8% higher digestibility. In other words, the biggest effects of the leaf stripping were on crude protein and, to a lesser extent, NDF. To address question 3, the higher crude protein and lower NDF concentrations in the leaf stripper fraction increase the feed value for monogastrics such as pigs. Pigs can utilize some amount of forage, and this can be beneficial for gut health. However, the relatively low crude protein content in the leaf stripper fraction is too low to be considered a protein feed in pigs. In addition, the high fiber content will limit inclusion in pig diets. The leaf stripper fraction could also be used to increase digestibility and crude protein content in the feed rations of lactating dairy cows, potentially reducing the need for grain-based concentrates. However, this needs to be confirmed with feeding experiments. The residual fraction with lower crude protein and digestibility could potentially be fed to dry dairy cows, heifers, or other ruminants requiring feed with less energy and crude protein. Given that organic farms in Northern Europe and Nordic countries often have high levels of red clover, using a production scale leaf stripping machine may be a valid option for producing high protein feeds, which could be preserved through drying or ensiling. The approach could be flexible, selecting leys with high clover content. In Sweden, it's most likely to be used in mixed leys with high clover content, for example, in the second or third cut. Thanks for listening to the presentation. You can read more about ProRefine at our website.